Okay, let's start with the overview of genotyping methods, and we'll look at various genotyping methods. So overall, genotyping methods, or also known as DNA fingerprinting methods, are the key molecular methods that are used in epidemiology. Okay. Unlike cloning or some protection assay or RNA inhibitors, there are many different molecular methods used in different disciplines, and immunology or some other viral or pathogenesis, other things. For epidemiologic purpose, the key ones are genotyping methods, which do have often, not 100%, not always, but often they do have better discriminatory power, better typability, better reproducibility than the traditional phenotypic methods that we use. Okay. So here are a list of the most common ones and that we will be covering as well. Pulsed field gel electrophoresis is very commonly done for foodborne pathogen genotyping purposes particularly, but it can be used for various organisms. Mainly not as small as virus, but larger ones, bacteria, fungus, and so on. We'll look at more details. Restriction fragment length polymorphism is another one. RAPD, all the acronyms you see here, AFLP, REP-PCR, MLST, MLVA, etc. So we'll see all these and others between today, and we'll continue on Monday. Okay. So let's see. So here is a quick description of what categories these methods are. Actually, you see the methods listed I just showed you on the last slide. What core approach are they based in? The original scientific manuscripts that showed their utility for epidemiologic purposes. So PFGE is the first one. It uses only restriction. It's a restriction based. There is no much of other sequencing, amplification, and other needed, but we'll see the details. It has been around since 1984. Shivars and his colleagues developed it for Saccharomyces cerevisiae, in fact for Saccharomyces, and then it was adopted for bacteria and some other organisms. Microarray is another method, also genome-based, similar to PFGE. I have another lecture on microarray. I don't think we'll be able to cover, maybe towards Tuesday if we have time, and we'll see microarray. It's a very good genome-wide microarray is used not just for epidemiology purpose, for lots of purposes, drug development, gene expression assay, for therapy, therapeutic purposes, and various other reasons. It has been around since 1995. Some methods are also hybridization based. One is restriction fragment length polymorphism. As the name indicates, definitely it deals with restriction, but there is also a component of hybridization. So hybridization is another core method, but less and less utilized, but we will cover it when we see RFM. And we briefly discussed it yesterday as well. Some of the genotyping methods used a random amplification. Perhaps the most common one used, in fact, more often in developing regions is RAPD, random amplified polymorphic DNA. RAPD is excellent in a way that it is very cheap, it is quick, you don't need to have a prior knowledge, but in terms of discriminatory power, others have some concerns, but we will see the detail later. Some methods do also based on amplification, but it's a targeted amplification. For example, amplified fragment length polymorphism. We will look at that. As the name indicates, it's amplified, so there is amplification. Fragment. So what other method do you think it uses among the colleagues? Restriction. So it's got amplification, restriction, and some other ligation in some steps. We'll look at that. Some methods, in fact many methods these days, use repetitive elements. There are some genetic components, whether it's human genome, a cow genome, a bacteria genome, there are these repeated elements in the genome. So some of the methods take advantage of those repeated elements to either sequence them and amplify them, and based on the fragment pattern or their pattern or amplification pattern, you can decide the genotype of isolate. Okay? So RAP PCR is here, but also we'll look at for TB purpose some other ones. VNTR, multi-locus variable number tandem repeat analysis, MLVA or VNTR is the same thing. And some methods are just sequence-based, multi-locus sequence typing, MLST. Okay, we'll look at MLST as well. All of these methods, regardless of what core methods they use, and of course our goal is mainly genotyping, knowing what type of strain or this infectious agent is, whatever organism we work on. But all of them show indirectly a variation in the sequence of that organism. Okay, indirectly. Or directly, MLST is the most direct one. It's multi-locus sequence typing. And as the name indicates, we look at multiple targeted areas within the genome, sequence it, compare, and establish a fingerprint. So we'll look at all of those, plus we'll look at oligotyping and some others. Okay. I know people are interested on TB mostly. Okay, let's start with PFGE, pulsed field gel electrophoresis. It is restriction digestion based, as I said earlier. In principle, it deals with an intact genomic DNA, of an organism, and using a rare cutter enzyme. Okay, it's a restriction based, it's intact genome, a rare cutter enzyme is important. What do I mean by rare cutter? Anybody can explain that? Okay, fewer fragments. Yeah. Okay, so if you have a rare cutter, what do you expect the recognition sequence of the enzyme? Larger, small, longer. Yeah. Okay. It is a longer recognition sequence and it cuts rarely. So we'll see the reason why and so on. And the separation of these fragments, because it is a rare cutter, the fragments are larger fragments and heavier in terms of molecular weight. Therefore, we have to use a unique type of gel. You cannot use just a regular horizontal gel to separate the fragments. Most of them are too large. Therefore, we use a contour clamped homogeneous electric field. It's a unique type of hexagonal shape electrophoresis machine, which I will show you. And anybody who have seen PFGE machine, do we have, does anyone, any laboratory has any piece or Ethiopia? Okay, maybe not, but it's not extremely expensive. It's much cheaper than a sequencer, but it's very useful machine. Maybe $40,000 or $50,000 you can get a complete set of PFGE machine. I haven't seen anybody's lab at least before. I don't know if HNRI or other has it, CDC maybe. Again, the principle is, okay, the DNA is forced to change direction during electrophoresis. That's why it is contour clamped. With this large fragment, you cannot horizontally from negative to positive, you can't separate. It's too large, too big fragments. Therefore, it needs to have this contour clamped, different direction type of pulsating. That's why it's called pulsed field, pulsating electric current. The upper size, it can really separate fragments as large as 10 megabase pairs and as small as 30 kb, 30,000 base pair to 10 million base pair size. Very large fragments. Unlike when we amplify a gene in PCR, it's 1 kb or 2 kb, and you can use small horizontal agarose gel to separate the fragments. Okay. And then there are different types of that, the electric fields, types. Let us know. How do we do it? Simply the methodology in a very simple way. The cells are embedded in agarose blocks. So this is one of the unique methods, as I mentioned to you yesterday, that you do not need, and you don't have to extract DNA. No DNA extraction. Fresh bacterial cells or fungal cells, whatever you use, not for virus, Viruses are too small to use PFG. No need to use PFG for virus. But fresh, pure isolates are embedded in agarose blocks directly. Okay. Of course, there is some steps in there. We measure how much the concentration of the cells, some technical details. And you can find that easily online as well. And then while they are embedded in agarose, the cell wall is digested, the cell membrane, cytoplasm, all the RNA, everything is removed except the genomic DNA. So that gives you an intact genomic DNA as shown here. So this is with the cells embedded after you do all the digestion while the cells are embedded in the agarose, you get the intact genomes. So while they are still in the agarose block, you digest it using a rare cutter enzyme. So this is already digested once, and now you can use the contour clamped homogeneous electric field or pulsating electric current to separate these large fragments, and you get similar gel patterns. So those separated gel patterns are, each one of them are fingerprints of, or just like a barcode that you see in different materials, just like a barcode you see different. So that's the fingerprint. It's the binding pattern is the fingerprint of the isolate. If you have 20 meningococcus isolates collected in 1 year period at Black Lion, you can run on a PFGE, look at their fingerprint pattern or their barcode and determine whether it is the same strain persisting in the hospital or is it a new strain coming from Jam, Sidamo, different places. Okay. So it's very useful genomic approach.
DNA is extracted, there is no extraction still.
No extraction. Yes.
The gene, there is something which extracts the DNA from the cell.
Okay, the question is, okay, there is no DNA extraction. In the gel, is there a DNA extraction? So it's more or less the reverse process. The DNA as well as the cell are embedded in the gel. You just remove the coating. Remove the cell wall, remove the cytoplasm, so the DNA will be extracted right there. It will be staying right inside the gel. You get my point? So it is extracted in a way, but you don't extract it in a conventional way of using mixing chemicals. Rather more a reverse process, just remove those other components so that just the DNA remains in the agarose. Okay. Or while it's in the agarose, you treat it with lysozyme, you treat it with detergent, just like we saw yesterday. You treat it with proteinase K, there is lots of washing process. In fact, since the cells are inside the agarose, it's more difficult. It's more involved. There will be lots of washing steps to remove the cytoplasmic dirt materials, the lipids, the carbohydrate, all those things still remain the same. The only difference is the cell is embedded in the agarose. You don't first extract and then put it in a gel or something. It's just the cell. Why is that? Why do you think you do it that way? What happens if we extract and continue the process without no agarose embedding? Did you see any keyword in the last few slides? That intact. Don't get an intact genome. If you extract DNA using pipettes, chemicals, there is a lots of shearing. You don't get good results. If you want to get intact genomic material, you embed the cells first in the agarose. Okay, clear? So, makes sense? This is mainly just to show you. This is the, I will not be able to see it well on your slides. You'll see it in the future. This hexagonal electrodes and the pulsating current machine. It releases lots of voltage, so there is a cooling module. It's a little bit different from regular agarose one. And you run the gel for about 24 hours, 18 to 24 hours, depending on the organism, depending on the type of enzyme used, and so on. Okay. So here is an example. So you see a fingerprint. I think I showed you yesterday when I when we discuss discriminatory power, just similar to that. So each of these fragment patterns you see, right? Each of these horizontal lines are the barcodes or the fingerprints of each isolate. Here is isolate designation, UAP, whatever, and then then you can add the information from antibiotic resistance pattern. All of these were multi-drug resistant ones, Salmonella, Serovarta, Ephemera. Other designations like phage type, they are definitive type 193, definitive type 104. They were collected from this, it is from a farm, pig farms. We collected from North Carolina in this case. Farm number, it can be a hospital, it can be environment, it can be a farm. Okay, but these are the fingerprint patterns. But what I haven't yet shown you, but hopefully Monday morning will cover, is you generate a dendrogram. Usually if you have few isolates, you can do it visually. Visually you can compare, right? If you see the first two or the bottom, some of them like this and this, they are the same, right? You can see the same pattern. Can you see same patterns or different? Yes? No? So visually you can decide which, if your question is on 2, 3, 4 or 10 isolates, is easy. You can decide. You don't need a sophisticated software. But often we do epidemiologic studies, so you may have 50, 100, many more and more isolates you have. It's difficult to compare visually. So you generate a phylogenetic tree. So what you see here is a dendrogram. Okay. So there is specific software, specific way to do that. We'll look at that on Monday once we finish all the genotyping methods. Okay. So this is a pattern from a PFGE. So then you interpret the data, and so what we saw is genotyping. It's not epidemiology. Right here, what I mean is, I showed you a PFG example, but your epidemiology question could vary. You might be studying transmission dynamics, you might be studying source of outbreak, you might be developing a surveillance system for a hospital or for a country. So all those epidemiology questions, you interpret your findings based on those informations. You will see more of those in terms when I give you examples of applications, okay, or the literature review. Hopefully, in fact, before we leave today, I will give you, okay. Any question on PFGE? Simple, straightforward process, but technically it could be a lot involving.